Good afternoon, everyone, and welcome to today's QAAFI seminar. My name is Bruce Topp. I'm a professorial research fellow with the Centre for Horticultural Science at QAAFI, and I'm based at the Maroochy Research Facility, where I lead the National Macadamia Breeding Program. I acknowledge the traditional owners and their custodianship of the lands on which we meet today. I pay respects to their ancestors and descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australia and global society. This seminar is scheduled from now, 12 noon, through till 1 o'clock. At the conclusion of the seminar, we will hold a question and answer session. If you have questions that you would like answered, please use the Q&A button at the bottom of the screen rather than using the chat button. We will address these questions at the conclusion of the seminar. So it's my pleasure today to introduce our speaker, Dr. Mobashwer Alam. Dr. Alam is a plant breeder and geneticist. He is currently working as a research fellow in QAAFI's macadamia breeding program. Previously, Dr. Alam worked as a senior plant breeder for Nuseed Pty Ltd. He achieved his PhD in plant science from the University of Queensland and worked on trait genomics, physiology, and modelling in sorghum. His research interest is to work on technologies to develop fast, accurate, and cost-effective tools for efficient breeding and for profitable orchards. Mo is based at the Maroochy Research Facility on the Sunshine Coast and is an integral part of our breeding team here at Nambour. Today, Mo will be discussing his work on macadamia rootstocks. And I'm not sure if you will mention as well, but on a side note, Mo is also in the middle of, in the process of building a house here on the Sunshine Coast, and I think has been very pleased to see that the site is well above any flood levels or damage from this past week of rain. So if he's got a smile on his face, that might be part of the reason. Anyway, it's my pleasure to introduce Mo, and I will hand it over to him now.
Thanks, Bruce. Thank you, everyone, for attending this seminar. And before starting my presentation, I would like to acknowledge the traditional owners and their custodianship of the lands on which we meet today, and pay my respect to their ancestors and their descendants. Thank you, everyone who are going to attend this seminar, and it is my great pleasure to present some research outcome from our rootstock breeding program. This presentation is a little bit summarization of multiple student projects in macadamia rootstocks. So before going on to my projects, I'd like to give you a brief outline why we are working on rootstocks. So commercial plantation of macadamia mostly requires clonal propagation through grafting or budding, and rootstock is the integral part of this clonal propagation system. In addition to multiplication for commercial plantation, rootstocks are also important for anchorage support with its long taproot system. As a long-lived crop, rootstock provide resistance to soil-borne pest and abiotic stresses, and ability to beneficially alter scion phenotypes, particularly for reduced plant size and early productivity. So rootstocks are important for increased productivity, orchard management efficiency, and sustainability of agricultural system. Macadamia has a long, prolonged juvenile phases and primarily outcrossing reproductive system, using genetically distinct rootstock with unique traits, both above ground and below ground components of the plants can contribute to the improvement of industry's productivity and profitability. A large number of rootstock studies has been conducted in other tree crops like apple and citrus, but only a handful of reports are available in macadamia. Initial investigation was conducted on 2 macadamia integrifolia scions grafted onto 5 tetraphylla and 5 integrifolia rootstocks. Results from these 2 studies suggested that tetraphylla rootstocks performed better than integrifolia rootstocks, but later Hamilton in 1988 suggested not to use tetraphylla but use integrifolia because tetraphylla may have latent incompatibility with integrifolia. In 1992, Trocolias reported the performance of 9 scion cultivars grafted onto 1 integrifolia and another tetraphylla seedling and cutting rootstocks. He reported that seedlings shows a higher early growth than cuttings, and cutting rootstocks are also prone to wind damage possibly due to not having a strong backroot system. In 2016, a preliminary investigation on 15 seedling and cutting rootstocks showed that rootstocks account for around 20% of the variation of scion yield and height. But it is still unclear how rootstocks mediate this phenotypic variation, and no comparison has been made on rootstock and scion phenotypes. To support the genetic improvement of rootstocks, Australian horticulture industry came through Horticulture Innovation Program and came forward and provided funding on rootstock investigation. As a part of macadamia improvement and conservation project, 15 rootstocks were investigated and preliminary result was published by Neil et al. In 2016. Another investigation on 203 rootstocks is ongoing to suggest high-performing rootstocks for the industry. For the first time in macadamia, we took the opportunity to focus on selecting rootstocks controlling plant size and yield efficiency. We are working collaboratively with these 5 industry-funded projects to explore the genetic, physiological, and molecular mechanism of rootstock effect on scion performance. In this study, we have used a diverse range of germplasms. Here we categorized into 5 categories: wild germplasm, cultivars, potential dwarfs, elite selection from breeding, and standard rootstocks. So this dendrogram shows the diversity of our rootstocks based on SNP data. We can also see how much genetic diversity covered in our rootstock trial. We compared with all the genotypes used in previous studies, all the 15 genotypes used in previous studies. Here are all the genotypes. And here we have 5 different groups, clusters from our study. In this study, we planted our germplasm in 2017 at Nambo. We have used only a single scion cultivar, which is non-precocious, called HAES741. And we have used 2 different types of rootstock seedlings, 20 seedlings and 14 cutting rootstocks. And the plantation was conducted using 5 incomplete bob design. Here is a little bit picture of our plantation. We have planted grafted and ungrafted. This is a grafted trees and this is ungrafted trees side by side. But we have followed a zigzag pattern, we call it staggered pattern. So ungrafted trees planted side by side of the grafted trees, but in one row it is on left side, in another row it is on right side, to reduce the errors of phenotypic variation. So this is the first study we conducted. We wanted to look at the effect of rootstocks on scion traits. A master's student's protocol is working on these projects. So we have collected phenotypic data from seven phenotypic traits, including total height, canopy width, canopy depth, rootstock height, skirt height, rootstock trunk circumference, and scion trunk circumference. We also got some derived traits from these data. We got scion height. We also got canopy area, canopy volume, and growth rate over time. Initial study shows that on early scion vigor shows that strong effect of rootstocks on scion traits, particularly scion trunk circumference, canopy depth, canopy area, and canopy volume. We also found type effects, seedling versus cutting effect on scion height, but we didn't see any rootstock type interaction for all these traits. Here is a little bit picture of rootstock effect on scion height. So overall we can see in most cases, cuttings shows high vigor than seedlings, which we didn't expect, but that is opposite to previous research findings from truck fields. On an average in cuttings, the scion height varies from 380 to 580, and similar variation we also have observed in seedlings. And these 3 cultivars shows that these 3 rootstocks show that they have a strong effect of reducing vigor, whereas H2 and one of our elite selections, they contribute to increase vigor in HAES 741. Only one interaction we can see in A268 in cuttings. It also contributes to increase vigor a little bit, but it contributes a lot in the seedlings contribute a lot in increasing vigor. Possibly due to the variation in the seedling progeny. We got similar sort of results on canopy area. Here you can see the standards, Biomon and H2, and these all cultivars, these all rootstocks reduced vigor of HAES 741, and these all rootstocks increased vigor of HAES 741 seedlings. And the variation canopy area varied from 2 square meter to 8 square meter in seedlings, and 3.8 square meter to 7.5 square meter in cuttings. A16 is a small cultivar, in current commercial small cultivar, and it shows that it reduced the canopy area of the 741. And one of our dwarf selection also contribute to the reduction of the scion canopy area. We have done another investigation to investigate on precocity and yield efficiency. Sophie Jones is a honor student who is working on that part, particularly on precocity part, with Lindsay Shaw and Christine Beveridge group. From initial investigation, we have identified that rootstock have effect on precocity. Here you can see CNIS, that means cumulative nut injury until 5 years of age. We also have yield efficiency, at 5 years age based on canopy area. We also have yield efficiency at 5 years age based on canopy volume. And we have found that rootstock have significant effect of cumulative nut and shell yield until 5 years of age. Here are the diversity of precocity, of effect of rootstocks on precocity. We have seen from 482 2 kilogram nut and shell yield due to the variation in rootstocks. And one of our selections, dwarf selections, that have low nut and shell yield, while one of our elite selection that had very nut and shell yield for seedlings. So it seems to us that until now, it's very early to say, but until now, we couldn't find any rootstocks that reduce vigor or increase precocity. We have conducted another study on rootstocks. We measure the stomata density on 3 rootstock cultivars. Sam Wakefield, he worked with us in a summer scholarship program. He investigated the aspect and rootstock genotype influence on leafy stomata density in macadamia species and published a conference paper. This is the effect of aspects. So we have 2 aspects here. One is north, another one is south. We know that in commercial field, north have very light intensity and south have low light intensity. So we have noticed that the stomata density is very high in north and stomata density is very low in south. The variation of stomata density varies from 300 to 360. In Arabidopsis, it was found that when we have high stomata density, the stomata size also is smaller. When we have low stomata density, stomata size is bigger, but we haven't investigated the stomata size yet. This slide shows the variation of stomata density in 3 cultivars, and one of our cultivar, WJMAS 2029, had highest stomata density, more than 350 stomata per millimeter square. And on the other hand, Biomont, that have near about 310 stomata density. Here in this picture, you can find distinct variation. This is the stomata density in Biomont, and this is stomata density in WJMAS 29. This is one of our elite cultivar. To find out the association of rootstocks with rootstock traits with scion traits, we investigated the correlationship. We conducted the correlations, and we found that strong correlation of canopy area, rootstock canopy area with grafted free canopy area. And another possible reason could be rootstock may have different vessels, and small vessels may regulate the water movement, and that can have influence on hydraulic signaling as well. In a previous study, Ben Toft conducted an investigation of variation in vascular system in 3 different cultivars, which varies in size. So Ben used 3 cultivars. One is D4, that is very high vigor. Another one is medium vigor, we call PB25, and another is a small vigor, we call dwarf B63. We found strong variation in plant height. Then we sectioned all 3 cultivars in the midpoint of the second internode of new flushes. We have used image processing tools to identify the xylem vessel size. First, we have defined different vascular system using 3 fluorescence filters. Then took out the pith, then took out the external areas. We only keep the xylem vessel tissues. Then counted number of xylems and measured the size of the xylems. Finally, we made a correlation between xylem size and plant height, and we have found a very strong correlation, and average vessel size explained near about 60% variation of plant height. To investigate the molecular mechanism of rootstock effect on scion, we have used our current knowledge in other crops. Over the years, several mechanisms have been proposed on rootstock-mediated vigour control. One of the proposed hypotheses is the regulation of water movement. Plant AQP play roles in water diffusion across cell membranes. In this mechanism, AQPs in root cell play a role as a gatekeeper and modify water conduction and gradients and impact on whole plant water flow and plant water potential. Long-distance signaling within plants involving aquaporin to coordinate water demand by the shoot with the supply by the water can help to explain this, this mechanism. Here on the left side, you can see the summary of root tissue signaling that occurs when roots are subjected to water limitation. The classic abscisic acid signaling is shown as direct link to the stomata through the hydraulic signal that release ABI in the shoot through an signal transduction process, which is still unknown. Also shown in the independent ABS signaling mode, where in bundle shape cell and or xylem parenchyma cells respond by reducing the activity of aquaporin. So we'll be using this knowledge if AQP play a role in reducing water supply in the root system, and then that can be involved in ABS signaling and stomata conductance. So this is another part. This is not we are considering at this moment. Another mechanism we are going to study through an honors project conducted by Sophie Jones, who is working with Christian Beverage Group. We'll be using our current knowledge of flowering and branching pathway in garden pea. So we know that auxin play a role in bud outgrowth through the inhibition of cytokinin and upregulation of stygolactone. So both can play a role in regulation of BRC, that's a branching genes, and that can help on that can play a role in bud outgrowth. In this study, Sophie will be studying if there is a relationship of AFT gene expression with a BRC gene expression. In addition to molecular mechanism, we are also collaborating with National Tree Genomics projects to identify the genes that have candidate markers that play a role, that have an effect on rootstock effect on scion. So we are on the process of extracting DNA, but we are working with Craig Hartner to identify DNA. Once we finish complete DNA extraction, then we'll be genotyping all the rootstocks. So then we'll also be employing genomic selection for rootstock effect on scion. So this is all about we are doing at this moment, and we focused mainly on the everground part, but it's still lot to do on the below ground part. As it is a tree crop, still we can't use any destructive method. We can't pull out the trees, but we can use some non-invasive root phenotyping technology to identify to explore the variation in root architecture in the rootstocks. That can be useful for future breeding program. We also need to work on resistant screening for soil-borne pathogen. And in future, if we can develop a rapid phenotyping tool for rootstock architecture, we can employ genomic prediction as well. Now we are facing some flood. Possibly also need to focus on breeding rootstocks for flood resistance, and also climate change is happening, temperature is increasing. We also need to focus on resistance against high temperature and drought. I would like to acknowledge the contributors. This project is jointly supported by the DF and University of Queensland and New South Wales DPI. A key element of this initiative has been co-funded by Horticulture Innovation Australia Limited, using the Across Horticulture Levy and voluntary contribution from DAF and matching funds from Australian government through the Hort Innovation projects, transforming tropical and subtropical crop productivity and national tree intensification. We are especially grateful to Hort Innovation, processors, growers, and other supports for this initiative. I would also acknowledge our team members here at NAMBOR and at UQ and in different DAF sites. Thank you very much.
Yeah, thank you, Mo, for that presentation. We have a few questions for you, but before we start on those questions, it might be useful for you to define yield efficiency. You mentioned earlier on in your talk yield efficiency. Could you explain that, please?
Sure. For yield efficiency, we consider two things. One is per unit area and per canopy volume. So total number nut in shell yield divided by per unit canopy area, and total nut in shell yield divided by canopy volume.
Thank you. Thank you. Okay, so one of the questions here is, were the control seedlings, the seedlings from 741, were the control seedlings from 741, or were they seedlings of the rootstock?
So we have two different type of rootstocks. So we have cuttings from the rootstocks. So we have seedlings from rootstocks. And we also have compared with the 741 cuttings as well. Yeah. But we don't have any 741 seedlings.
Good, thank you. Another question is, would you expect the scions to differ in precocity if they are from non-juvenile material?
Yes, yes, I would expect, because we know that if you collect scion from one side of the tree and another type of scion, you can see some variation in plant architecture, certainly. And for precocity, that may not have large effect because the whole plant is not in juvenile stage. Whole plant is in reproductive state. So possibly you may not see that much variation for precocity.
Thank you, thank you. And another question here about stomatal density. And the question is, stomatal density, is this mainly related to canopy shading, with denser canopies having lower density of stomates?
Yes, that's happened. So that's why we have collected stomata from two sides, one from external north side and another from external south side. So external south side got density, but we have found genetic difference as well in both sides.
Good, thanks, Mark. And yeah, another question here from John Chapman. Do seedlings generally produce true to type?
No, no, because macadamia is highly heterozygous, so seedlings are often pollinated, so it can get any pollen from any cultivar. So they are not true to type. So you can see a lot of variation in seedlings and less variation in cuttings.
Thanks, Mo. And John also makes the comment, congratulations on the ongoing rootstock work, and he hopes the funding continues. So that's some very well appreciated comments there. Mo, I've got another question for you. The Hamilton paper that you mentioned that had they encountered the problems with tetraphylla with the compatibility. In the propagation work you were doing for this rootstock trial, what was your findings with regard to that?
No, we haven't seen that incompatibility. So we have used 2 tetraphylla in our study. So we haven't seen that much incompatibility. But we have seen some incompatibility with the Jensen I. So the success rate was also low when we grafted. Yeah, especially when we have used cuttings.
Good. Thanks, Mo. Well, look, that's the finish of the questions. I just want to let everybody know that, first of all, thank Mo again for the presentation. And I'd like to let everybody know that the next science seminar next week is International Women's Day, and there will be an International Women's Day seminar on Tuesday, the 8th of March, and it's titled Panning for Genetic Gold, Pan-Genomics and Other Omics Resources for Crop Improvement, presented by Dr. Emma Mace from QAAFI's Centre for Crop Science. So that's next week. And thank you, everybody, for attending our science seminar today. If you haven't already signed up for the seminar invitation list, please do so. And thanks once again, Mo. And thank you, everyone, for attending.
Bye, everyone.